Good day, everyone. My name is John Mills. I'm a senior staff scientist with bioMérieux. Today, I'm going to be discussing with you the impact of AOAC for cannabis labs and lab validations. So what will you learn today? What are we going to talk about? Who is AOAC and what is the role of AOAC? How does AOAC ensure and impact cannabis science and safety? What is the value of AOAC certification for the cannabis industry and you? What does this mean for you in the cannabis industry? And how does AOAC impact state-required validation? So first, who is AOAC? AOAC basically establishes standard methods of analysis. These standard methods of analysis impacts health and safety, and they are globally recognized. If you look at the AOAC webpage, they have some information on their legal name. Their legal name is AOAC International. The long form name is Association of Official Analytical Collaboration, AOAC International. And their new tagline has evolved into "In Food and Agriculture, we set the standard." The organization name has evolved throughout the years, and we will discuss this further in the presentation. There are three main programs: the Official Methods Program, the Research Institute, and Proficiency Testing. Of note, with the Official Methods Program, OMA methods are recognized in the U.S. Code of Federal Regulations, and they are legally defensible in court worldwide. Taking a look at AOAC's mission, as a leader of analytical excellence, AOAC International advances product safety, product integrity, and public health by bringing together members, organizations, and experts dedicated to developing and validating standards, methods, and technologies of global relevance. Going back and looking at the AOAC roots in product safety, AOAC initially began as the Association of Official Agriculture Chemists in 1884. Federal and state departments of agriculture, through the Bureau of Chemistry, initially was formed to standardize methodology to be used for composition of fertilizers by state laboratories, formed the original AOAC. This was directed by Harvey Washington Wiley, who wrote the 1906 law that began the U.S. Food and Drug Administration, FDA. Through a number of government reorganizations, the Food and Drug Administration came into being as a result of the Food, Drug, and Cosmetic Act of 1938. It became separate. The AOAC was housed within the FDA and became the Association of Official Analytical Chemists. For the next 40 years, only government chemists could become members. In the 80s, membership evolved and included microbiologists, food science scientists, professionals. In 1991, Association of Official Agriculture Chemists legally changed its name to AOAC International. Association of Analytical Communities has been used to encompass all of the scientific disciplines involved in AOAC's work. So here's some additional details regarding AOAC and its current activities. We talked about the main focus of AOAC is standard developments. We have two different programs for that: standard methods performance requirements, SMPRs, which we will talk about quite a bit during this presentation, official methods of analysis. Also, if you look at government and organization, there is a membership elected board of directors, and there's also official methods board. For membership, it's around 3,000 individual member scientists, organizational affiliates, 16 global AOAC sections, multiple analytical communities. There's awards and recognitions which are given at the annual meeting each year, which includes going back to the founder, the Harvey Wiley Award. For laboratories, there is a accredited laboratory proficiency testing program. There's also laboratory auditing that is available. The ALACC guidelines, which is accreditation guidelines for laboratories, this is AOAC accreditation guidelines for laboratories, is also available. ALACC is a multinational group of experts from a varied scientific disciplines and industries and part of AOAC's technical division for laboratory management, or TDLM. 3 publications from AOAC, that's official methods of analysis of AOAC International, and there's around 2,800 methods in the official methods of analysis. There's a Journal of AOAC International, and also Inside Laboratory Management magazine. We also have the Research Institute, which is a division of AOAC. That's where we have validation protocol development, performance tested method certification, and official methods of analysis program when it is harmonized with the PTM, and we'll go through this quite a bit later in the presentation. So this is a representation of how AOAC fits into product safety for really any industry. There are multiple layers to product safety within a manufacturer. If you look at the graph on the left, you see manufacturing process on the outer ring. You go into HACCP, the hazard analysis critical control points. From there, critical control point and hazard evaluation. And then at the center is of this is analytical methods. This is where AOAC comes in. AOAC helps prioritize analytical needs, creates programs such as CASP, which we'll talk about quite a bit, to develop SMPRs, validates methods for those SMPRs. These methods are then published and available to the industry. So in the end, the industry will have validated fit-for-purpose methods that can be confidently implemented. So here is a world picture of how AOAC leverages the power of many. AOAC uses its global network to gather stakeholders and experts with the mission goal of providing fit-for-purpose methods as discussed in the previous slide. This map details the locations of AOAC stakeholders and AOAC sections throughout the world. So as you can see in this really quick snapshot that AOAC is represented nicely worldwide. So how does AOAC influence cannabis safety, or science and safety? This is through the CASP firstly. I would say that CASP is a fantastic example of how AOAC influences cannabis, and that stands for the Cannabis Analytical Science Program. As detailed here, CASP is a forum where the science of hemp and cannabis can be discussed and cannabis standard methods developed. The CASP group has been working very diligently on standard methods development and have created several, which we will discuss more in the next slide. Also, experts in science and validation. AOAC and CASP have been able to bring together cannabis experts, validation experts, and technology experts to develop critically needed standards for cannabis. A little bit more on CASP. If you look at the scope and the objectives, of course, it's develop SMPRs for cannabis and hemp, extend a call for methods for each of the completed SMPRs, impanel an expert review panel to review candidate methods with the goal of delivering consensus-based first action and final action official methods. You see below the different working groups, you have microbiological contaminants, chemical, cannabinoids, training and education, and also proficiency testing, which we know is important for the cannabis industry. I actually pulled this information off of the AOAC webpage, aoac.org. There is a section under Scientific Solution tab where you can find more info on CASP if you'd like to research for yourself. What does AOAC bring the cannabis industry? So firstly, method testing validation. Third-party independent evaluation of methods is very important. Reliable analytical test methods. We talked about fit for purpose. These AOAC will allow you to have fit for purpose methods which meet your needs and what the industry needs also. And again, ensuring science and safety. This is a rigorous validation of analytical methods and gives you confidence moving forward with those methods. And at the end of the process, this collaboration, this is what we want to accomplish. Methods that help protect the industry, consumers, and products. So the SMPR performance requirements. Here's just a quick overview. I have a couple slides on this just to show you the process. Again, SMPRs articulates a community's analytical method need, fit for purpose again, build on the fitness for purpose. A very detailed description of the analytical requirements, includes method acceptance requirements, includes study designs for measuring analytical performance, used to qualify methods for AOAC approval. And these are published as a standard. You can go on to aoac.org and pull these up. They are open to the public. This process has been especially critical for the cannabis industry as there were no available standard methods to compare to for validations and verification studies. The CASP group has been able to collaborate and create the yardstick for which any method must be compared to and produce equivalent results. So another quick overview of the SMPR process. The draft SMPR is created by a working group for cannabis that would, of course, be CASP. The stakeholders review and comment. So after the working group develops the SMPR, they're sent out for comment. After the comments come back, the stakeholders review that. You have a consensus on the draft standard, and then the final standard is prepared for publication. Again, you can see that on the AOAC webpage if you go to the CASP tab. So on that CASP tab on the AOAC, I did pull off the list of standard method performance requirements that the CASP group has been working on. You can see there's 2 on cannabinoids, detection of aspergillus, detection of salmonella species, detection of shigatoxin producing E. coli, and determination of heavy metals. There's also a draft SMPR for yeast and mold enumeration that has been sent out for comment. You can see by looking at this, the CASP group has definitely been busy. One other program I did want to highlight before we get into some specifics regarding AOAC validation processes is the emergency response validation, or the ERV. The ERV validates existing PTM approved methods for new foods involved in emerging product safety crisis. This was initially the goal of the ERV, just to validate current PTM approved methods, but AOAC has expanded that to new PTMs, and a good example of that is the COVID environmental ERV that happened in 2019, which provided a PTM approval for methods which did not currently have a PTM approval. It's also a nice program because it's fast-tracked. This can take less than 3 months. We'll talk about the timing of the other programs later in the presentation, but this can take less than 3 months. AOAC arranges the preparation and distribution of the test samples. They provide step-by-step support. Each successful candidate method is granted a certificate of validate, and then the results are published in Inside Laboratory Management, the Journal of AOAC, and on the AOAC RI website, again, that anyone can access and pull that information off. These projects are normally determined by AOAC management based on global crisis or gaps in food commodity safety testing. And this program was actually recently employed by AOAC for an ERV in cannabis, and that's yeast and mold enumeration. Let's talk just a little bit about AOAC Evolution and the OMA program and the Research Institute. So the Official Methods of Analysis program actually initially was not designed for commercial methods. AOAC International developed the Performance Tested Methods program for those test kit manufacturers, created the AOAC Research Institute to run the PTM program. So PTM programs specifically to evaluate and certify commercial test kits. And this allows a harmonized approach with a PTM and an OMA, and we'll talk about this just a little bit later. This is an efficient process and appeals to the industry. The reason it appeals to the industry is that prior to the Research Institute and performance tested methods, if a test kit manufacturer or anyone wanted to have an official methods analysis, they would have to go through a pre-collaborative study first, and then the collaborative study. We will go through this information a little bit later, but the RI in the PTM allows you to have a certificate almost like midway through the process, so you can have a certificate before you go to official methods of analysis. It also allows you to have an approval or a certificate if for some reason you may not be able to have enough collaborative labs. And so it's a nice middle point that gives you a PTM number, gives you a proof source, and as it's mentioned here, it is profitable to AOAC. This is a real quick snapshot of everything that goes into an AOAC validation, and we'll talk about all of these: inclusivity, exclusivity, matrix study, stability, and robustness. The core of the validation is definitely the method comparison study. The remainder of the validation, we go back to inclusivity, exclusivity, shelf, ruggedness, etc., are all still very important. However, the method comparison is where a method will demonstrate that it is equivalent or better than a reference method. Here's a quick snapshot of what that looks like. You spike matrix and you test both the alternative method and the reference method. For cannabis, this would be compared to an SMPR that came out of the CASP group. So more information on the matrix study itself. A selection of matrices associated with the pathogen. Which matrices are most important to the industry? For cannabis, this is of course would be flower and hemp. Other matrices can be added after initial approval via matrix extension if needed. We probably will see this in the cannabis industry as more and more matrices become important. Natural contamination is always preferred. However, at times it can be challenging, so artificial contamination is permitted. Which strains are important to an industry? For cannabis, this is a little difficult at the moment, but data are being collected daily from cannabis laboratories and other experts. We will know more at some point. Competing flora. Is it important to include competing organisms? Will they normally be present in addition to the target organism? This could potentially be important for cannabis for sure, just because we know that there is a significant bioburden with flower and hemp. And lastly, fractional results. This is artificial contamination at a level where only 25 to 75% of the samples are positive with either method. This theoretically ensures that some samples will be inoculated with only a single cell. Pathogen screening methods need to demonstrate that one cell can be recovered and detected per test portion. That is critical. Here's an example of matrix study workflow. The validations for cannabis will be a little different because they are paired. However, this will give you a good overview of what is normally executed. A product sample is spiked. Again, you will have a fractional or high level. The candidate method will be tested on that test portion in parallel with the reference method. You'll perform the assay, the candidate method confirmation. For the candidate method enrichment, you will also do a secondary enrichment or an extended enrichment. And then from that, you will do the reference method for confirmation. And then this is compared to another test portion which completely followed the reference method for that assay or target. So a little bit closer look at both the PTM and the OMA, what is needed, what is required, what is normally done. AOAC performance tested PTM methods. You need to have one independent laboratory. And for each matrix, you test 20 replicates at a low spike to LOD. We talked about fractional. Those need to be at the fractional level, so 25 to 75% of samples are positive. 5 high spike level replicates and 5 uninoculated. When we move to the OMA, this is a multi-laboratory study, a minimum of 10 labs. We normally do at least 15 because you're looking at, you need 10 good data points. Prereq for this is either a PTM or a pre-collaborative study. The collaborative study is an interlab reproducibility. Same spiking criteria as a PTM, but there are fewer replicas, just more overall data points because you're looking at multiple laboratories. So a little bit more about an OMA validation process, and we talked about how the OMA process has evolved, because at one point you had to do a pre-collaborative followed by your collaborative. You didn't have the choice to do the PTM again, which is a midpoint. You didn't get a certification at that midpoint. So if we go back to the old school pre-collaborative study, is performed by one laboratory. This is a contract laboratory. All matrices claimed are evaluated at that lab. And, again, it differs a little bit, is that if you follow harmonize, that would need to be an independent laboratory. Phase 2 is the interlaboratory collaborative study, so multiple laboratories. That follows exactly what we talked about before if you went from a PTM to an OMA. So a little bit different, and you can go direct. bioMérieux has done that in the past where you skip the PTM process, you collect pre-collaborative data and go directly to an OMA. So it can be done, it still is done, it matters on. if you want the certificate and timelines, etc. More information on the performance tested methods. We talked about each of these just a little bit before. Inclusivity, exclusivity is required. Method comparison. You need to test at least 10 products for a variety of foods. Since we're talking about cannabis, I wonder if we may discuss with AOAC about changing that to a different term. Right now, that is what they would call a variety of foods for your performance claim. And then you need to determine the correct reference method. Of course, for cannabis, we'll be talking about SMPRs. You need to demonstrate ruggedness. This is looking at package insert parameters just outside of what is in your package insert to demonstrate that your system is rugged. Stability is shelf life. You need to provide data to demonstrate your shelf life. And on top of that is that, do you see any variations when you look at multiple lots? At the independent lab, it's just a single laboratory and a minimum of one product tested at that single laboratory. Closer look at inclusivity. This is being able to demonstrate that an assay has the ability to detect target analyte from a wide range of strains. So you need to test a minimum of 100 different serotypes, for example, for salmonella, that's out of the greater than 2,200 serovars, and a minimum of 50 for all other organisms. Exclusivity, you need to demonstrate that there's no interference with your assays with non-target organisms, and that's with a minimum of 30 isolates. Now, there is always a balance between sensitivity and specificity when you're talking about near neighbors and different types of assays. The priority will be given to sensitivity there. Official methods of analysis, we did talk about this just a little bit. You can either do a harmonized approach with a PTM or a pre-collaborative. But if you did the pre-collaborative study, you do need inclusivity, exclusivity, method comparison. Normally, we would have about 15 to 20 different products, and compared to a reference method. We have USDA, FDA, and ISO listed here, but for cannabis, it would definitely be in the SMPRs. And then go to the collaborative study again, 10 labs minimum, a minimum of one product, and then a reference method. So how long do these normally take? If you look at official methods of analysis, this is normally 12 months for approval. AOAC Research Institute is 6 months for a PTM approval. And if you do the harmonized approach, again, where you would get a PTM first and then move to an OMA, that's normally 12 months. I will say, though, that AOAC has made a focus to decrease these times. And I think if we went back and looked in the last 2 or 3 years, all of these times are going to be decreased because they did put, there was a huge focus in dropping these. So I'm not sure these are quite accurate because AOAC does a good job of turning everything around, and the volunteers also do a great job of that. We talked previously about some of the information that you can get off of the AOAC webpage, and they also have a nice function that you can go in and search all of the AOAC PTM validated methods. So if you go into the AOAC PTM tab, and there is a searchable area. I have that pulled up here. This is just a screenshot from their webpage. And you can search based on the analyte, the matrix, the manufacturer of the test kit, the kit name, or the PTM certificate number, the license number. So here's an example. I went into the website and I searched for cannabis, and here are the 3 methods that came up that are currently AOAC PTM approved. That's the GeneUp Salmonella, the GeneUp EHEC or STEC, and the GeneUp Aspergillus Pro. So let's go back to some of the questions that we had at the beginning of the presentation. Is that why is AOAC important? We talked about leveraging the power of many. AOAC unites industry, academia, and others. There are a lot of great resources in all of these different aspects that we can bring in and help each other and create methods, standard methods, SMPRs, working groups, etc. Established method testing validation. We talked about that, a key role of AOAC. In the end, AOAC confirms reliable analytical test methods. And of course, that then circles around to ensuring safety of consumers and this impacts public health throughout the globe. So going through AOAC, working with AOAC closely, why is there value for companies like my company, bioMérieux? So AOAC provides a rigorous third-party, independent, which is important, validation of all of our methods. When we have AOAC approvals, it gives an immediate level of confidence for our users, our customers, our potential customers. It also helps our customers when they move toward ISO accreditation for a laboratory. There are some regulatory agencies and programs within those regulatory agencies that require an AOAC approval. It's also nice that an AOAC approval gives us a data package, gives us proof sources. So when we go through a PTM, when we go through an OMA, our users can go onto the AOAC webpage, pull up the summary for the RIPTM. They can go to the journal and pull up the OMA article also. So great proof sources, data packages for our customers that demonstrate the performance of the assays. And we do work with certain companies that have a lot of them are larger companies, but they may have an internal requirement that they will only use AOAC approved methods. And we actually do have some that require that they will only use an OMA method. What is the value of AOAC certifications to you? So when you're looking for an analytical method in your lab, when you're looking for a method for any analyte that you have or target that you have, if you go to AOAC and consider those, what have been approved, those methods may have enhanced performance compared to others on the market that have not gone through a rigorous third-party validation. The value also is that rapid methods can be validated, a third-party validation. It is independent as listed here, which is always important. AOAC will validate all claims in the package insert. This is especially important in the PTM process, which goes through and you must validate any claim that you have in your package insert. All test kit manufacturers must meet a certain level of performance standard. And we brought up the word confidence before, but this gives the user confidence in the test kit, the analysis method that they are using. Brand protection is really important in any industry. It's going to be important in the cannabis industry also. And again, this goes back to confidence. You have confidence with AOAC approved methods. These are third-party, rigorously validated methods, so you can have the confidence that these methods will protect your brand. And lastly, I have noted here certificate of analysis. We do have customers that will list our AOAC, either PTM or OMA methods on their certificate of analysis to share with their customers, their laboratory customers. How does an AOAC certification influence lab validations? So I think we talked about this quite a bit, but AOAC validation demonstrates the performance and the rigor and the fit for purpose of the method. When these methods come out with AOAC approved or an AOAC approved certification or an OMA, they're basically plug and play. They're ready to be moved into a laboratory. Many times, depending on what the analyte is or the matrix is, if you have an AOAC approval, you can go directly into a lab with only verification. So you don't have to revalidate those on site. You just do a simple validation or verification. ISO accreditation. These methods, if they have an AOAC, can help you during this process. To what extent you need to talk to your accreditation, company that you're working with, but it can be benefit moving forward. And then kind of an open question, but an interesting one, is that how is this going to impact cannabis and state regulatory acceptance? So our key takeaways. AOAC and cannabis is the perfect collaboration. This is an emerging market, and working with AOAC is going to be a huge benefit for an emerging market. Standardized testing, collaboration with experts outside of cannabis, it's just a huge benefit. We talked about SMPRs quite a bit. They are going to be the yardstick moving forward for new method comparison in cannabis. AOAC approvals benefit both the test kit manufacturers and the users. We discussed this. And then lastly, AOAC is a fantastic resource for the cannabis industry moving forward. Thank you for your attention. please reach out to aoac.org for any information about the CASP group or any PTM approvals for cannabis and hemp. You can also reach out to me directly with any questions. Thank you again.